All right, I'd like to welcome everyone to today's edition of the Polyploidy Webinar, or some folks call it the Ploidy in Place series. Today we have two excellent speakers. Up first is Anne Curé, who is currently a PhD student, or PhD candidate, I should say, at Syracuse University. Anne has a BS in Integrative Biology from the University of Illinois at Urbana-Champaign. She is a PhD student right now in Carrie Seagrave's lab, where she studies the effects of neopolyploidy on plant-herbivore interactions. And she's going to tell us a little bit about that work today in her talk on neopolyploidy and Arabidopsis thaliana affects performance on the generalist insect herbivore Trichoplusia. With that, I'll let Anne take over and tell us about her really interesting research.
Thanks so much, Mike. So hi, everyone. I'm really excited to tell you about this work today. Before we start, I'd like to take a second and acknowledge all of my co-authors who couldn't all be here today. Most of the people that you see on this screen are actually undergraduate students who were part of a Cure-style course held at my university in the spring. And they were all instrumental in this work, and I'm so excited to be able to tell you about it today. So plant-herbivore interactions are some of the most influential interactions in our ecosystem. In fact, herbivores alone can remove about 20% of above ground biomass annually. And herbivores, in turn, are really affected by multiple traits within their host plant. In particular, herbivore performance, by that I mean their growth, development, or survival, are really affected by these traits in their host. So for example, chemical defenses can alter an herbivore's survival or development. Similarly, physical defenses such as trichomes and thorns can also have a really large impact on herbivores. Also, a plant's nutrients has an impact on how herbivores develop. And similarly, even traits such as leaf toughness could have a large impact on herbivores trying to feed on a plant. And in turn, all of these effects can also be impacted by cross-generational effects. And by that, I mean effects passed on from a parent to their offspring. Sort of overall, all of these traits can also be impacted by the local adaptation of the plants that we're studying. And one of the reasons that I'm really interested in looking at how whole genome duplication can alter plant-herbivore interactions is because we know that whole genome duplication can alter these important traits for herbivores. So for example, we know that whole genome duplication can either increase, decrease, or even keep chemical defense production the same. Even physical defenses can change. So this example I have shown here is trichomes on diploid and a tetraploid plant. It might be kind of hard to see, but the diploids are three-pronged, whereas the tetraploids are actually four-pronged. And that could have important ramifications for herbivores trying to feed or move on a plant. Similarly, there's also been work which has shown that polyploids may be more nutrient limited than diploids, and that can have an impact on herbivores feeding on those plants. And even components of different ploidy level cell walls can change. So in this graph I'm showing you, it's measuring lignin and cellulose content in 4 different cytotypes of plants. And what we find is as ploidy level increases, these compounds decrease. And this could make it easier for herbivores to feed on these plants. However, one thing we don't know is how cross-generational effects could change these traits in polyploids. And the reason that we think that cross-generational effects might be important is because we know that cross-generational effects can impact herbivore performance. So in this example from Rassman et al. 2012, the authors were looking at larval mass or performance on the offspring of undamaged or damaged plants of Arabidopsis thaliana. And what they found is that the larvae developing on the offspring of the undamaged plants, represented by the white bars, weighed more or were performing better than larvae developing on the offspring of the damaged plants. So again, highlighting that cross-generational effects were altering their performance. And also one thing that's been abundantly clear from the whole genome duplication literature is that local adaptation plays a really important role in altering plant-herbivore interactions in polyploids. A great example of this comes from Halverson et al. 2008, where the authors were measuring herbivore attack rates on the 3 different cytotypes of Solidago altissima. And for this fly species, for example, what the authors found is that the fly was attacking the tetraploids most in every population. However, for this moth species, what they found is that in one population, the moths were attacking hexaploids more, while in another population, they're actually attacking tetraploids more, showing that the geographic origin of our polyploids can alter plant-herbivore interactions. Similarly, Hull-Sanders et al. 2009 found that herbivore performance could change on polyploids from different geographic origins. So in this case, the authors were looking at herbivore performance on the 3 different cytotypes of Solidago gigantea from the US and from Europe. For this moth species, they found when they looked across cytotypes that the moth had similar weights or was performing similarly. However, when they looked within tetraploids, they found that larvae were developing worse. They had lower mass on tetraploids from the US, represented by the black bar, as opposed to larvae on tetraploids from Europe, represented by the gray bar. However, when they looked at the performance of beetle larvae on these same plants, they found that performance didn't change at all based on cytotype or based on geographic origin. And this leads us to sort of the big question that we're still trying to answer, which is how does whole genome duplication impact herbivores? So on one hand, whole genome duplication may impede herbivores, thereby creating a barrier to herbivory, while alternatively, whole genome duplication could be benefiting herbivores and therefore act as a mechanism for host expansion. All of our past works of whole genome duplication and herbivory have been done in established tetraploid populations. And one problem that we face when measuring herbivory in these populations is that established tetraploids have, of course, undergone a whole genome duplication event, but then following that event, they've also been subjected to potentially thousands of generations of selection. So one problem we face is how do we determine if it's selection or whole genome duplication itself that's actually altering the changes in herbivory that we're seeing? And one way we can address that question is by using neopolyploids, or in this case, neotetraploids. And the advantage of using a neotetraploid is that while they've undergone whole genome duplication, they haven't been subjected to all of those generations of selection. And this is really nice because it allows us then to directly test the effects of whole genome duplication on herbivory. So my first question for today's talk is, how does neopolyploidy affect herbivore performance? And does the geographic origin of the different cytotypes affect herbivore performance? To answer this question, we use diploid, neotetraploid, and an established tetraploid of Arabidopsis thaliana. Arabidopsis thaliana is an autopolyploid, and we were able to buy seeds, and autopolyploids are, Arabidopsis is a great organism for herbivory studies because it's a fast-growing annual, and we know a lot about its genetic background as well as its different types of defenses. And for our herbivore, we use Trichoplusia or cabbage loopers. Cabbage loopers are generalist herbivores, so they feed on many plant species, including Arabidopsis thaliana. They're really easy to buy and they have a very quick life cycle. And for all of these reasons, they're commonly used as herbivores in studies of herbivory on Arabidopsis thaliana. So today we use diploids and neotetraploids from 3 different ecotypes or geographic locations of Arabidopsis, shown here on the map. And then we also used one established tetraploid population. So to answer our first question, we first obtained diploid and neotetraploid seeds. Our neotetraploids in this study were created by Luca Comai and his colleagues. They took a diploid plant, dipped it in colchicine, and then collected both diploid and neotetraploid seeds off of that plant. So the diploids and the neotetraploids in our experiment are directly related within ecotype. So we then grew up those plants and then put individual larvae on the plants. And then to address how geographic origin could alter herbivore performance, we did this on 3 different ecotypes listed above in the green boxes. We then kept our larvae and our plants in a growth chamber. We contained larvae on plants using these homemade insect cages made from transparencies and barbecue skewers and rubber bands. And then throughout the experiment, we measured pupil weight, days to adult or development time, survival, and composite fitness. And we calculated composite fitness by multiplying pupil weight by survival and then dividing that by development time. So for our results, I'm first just going to orient you to our graphs because I'm going to show you a bunch of graphs that have a similar structure. Across the x-axis, we have our different ecotypes. And then the orange bars represent larvae feeding on diploid plants, whereas the blue bars represent larvae feeding on the neotetraploid plants. And what we found is within the ecotype AUA, larvae had higher fitness or were performing better on the neotetraploids compared to the diploids. However, larvae feeding on diploids from Prudka instead had higher fitness or were performing better than the larvae on the neotetraploids. Similarly, when we looked at pupal weight, we found the same trends, where in AUA, the larvae were performing better on the neotetraploids, as opposed to in Prudka, where the larvae were performing better on diploids. When we looked at development time, we didn't find that development time differed at all between the cytotypes or the ecotypes. And we found a similar result when we looked at survival. So the larvae survived equally on all of our different plants. And from this, one conclusion that we draw is that it doesn't appear that whole genome duplication is creating a barrier to herbivores. Our herbivores are still able to survive and feed on plants, maybe though they're performing differently on those different cytotypes. And what we really saw was that the geographic origin of our neotetraploids is having a major impact on our herbivore performance. Also, for this specific comparison, it looked like pupal weight was really driving the trends that we were seeing. Because again, since composite fitness is made by multiplying and dividing our different response variables, and since pupal weight is one of those, it seems like pupal weight is driving that trend we see in composite fitness. So next, I'm going to answer the same two questions again, but this time I'm going to compare herbivore performance on our 3 neotetraploids and on our established tetraploid. So the reason we decided to keep the established tetraploid out of our comparison with the diploids and the neotetraploids was because we don't have a perfectly balanced design. So we don't have established tetraploids from every ecotype. And therefore, so we can avoid potentially confounding our results, we decided to just keep them separate and instead just compare all of the tetraploids to each other. So again, I'm going to show you a series of graphs that are all going to look similar. Across the x-axis is our ecotypes. The gray bar represents larvae feeding on established tetraploids, whereas the blue bar represents larvae feeding on the neotetraploids. So for composite fitness, what we found is that larvae on the established tetraploid had a similar fitness or performing similarly to larvae feeding on neopolyploids from AUA. Then the larvae on neotetraploids from C24 were sort of in between. And again, our larvae from Prudka had the lowest fitness, so they were performing significantly worse than all the other larvae. For pupal weights, we found that our larvae on the established tetraploid had similar weights or were performing similarly to larvae on the neotetraploids from AUA and C24. But again, our larvae from Prudka weighed significantly less and therefore were performing significantly worse than the larvae on all of the other tetraploids. So for development time, we found that the larvae on the established tetraploid actually developed the quickest, so they performed the best, followed by the larvae on the neotetraploids from AUA, with then the larvae on the neotetraploids from C24 and Prudka performing the worst. And then again, when we looked at survival, we found that all of our moths survived equally on our tetraploids. So from this section, we conclude that our larvae perform similarly or better on the established tetraploid compared to the neotetraploids. However, again, we're seeing that the geographic origin of our neopolyploids is again having a huge impact on herbivore performance. So for my final question, I wanted to know if cross-generational effects and polyploidy can interact to impact performance. So to answer this question, we had the same general design as we did for the first two questions, except this time we had a maternal generation where our plants were either subjected to herbivory or were not. And then we collected seeds from those plants, and then the offspring are what we used to actually measure herbivore performance on. So again, I'm going to show you graphs that are all going to look pretty similar. Across the x-axis as always is ecotype, except this time the white bars are going to represent the larvae on the offspring of undamaged plants, whereas the black bars represent larvae on the offspring of damaged plants. And for composite fitness, what we found is that The larvae perform best or had the highest fitness in C24 on the offspring of the undamaged plants, as opposed to larvae which were on the offspring of the damaged plants. However, in Prudka, we see the exact opposite as always, where the larvae were performing worse on the offspring of the undamaged plant, as opposed to larvae which were on the offspring of the damaged plant. For pupal weight, we again saw the similar trend, where the larvae on offspring of the undamaged plants had higher weights or were performing better than those on offspring of the damaged plant. And in Prudka, we saw the opposite, where the larvae were performing better on the damaged or the offspring of the damaged plants, as opposed to the offspring of the undamaged plants. Finally, when we looked at development time, we found that development time didn't differ at all based on cytotype or ecotype, or really our cross-generational effects. So some conclusions. One thing I forgot to mention was that to analyze this, we used a three-way ANOVA, where we looked at the effects of cross-generational effects, cytotype, and ecotype. And we never found a significant interaction between those three. Instead, what we found was we primarily saw that cross-generational effects and ecotype instead had a significant effect on herbivore performance. And we didn't find that cross-generational effects that cross-generational effects altered herbivore performance at all on either cytotype. So again, the main theme that we're really seeing from this is that the geographic origin of our polyploids is really important. So some big takeaways that I'd like you to take from today's talk. I feel like I've said this so many times, but again, the geographic origin of our polyploids is going to play a vital role in influencing herbivore performance on these polyploids. And I think this is really interesting because since we know polyploids can arise repeatedly within populations, and since we also know that polyploids with different geographic origins are going to alter how they interact with herbivores, then this in turn could all suggest that polyploids can also alter plant-herbivore interactions in their community, which I just think is fascinating. And then from this, it appears that whole genome duplication may not necessarily act as a barrier, at least in these populations, but instead it may actually act as a selective force. So by that I mean that whole genome duplication could almost act as a diversifying agent for the insect herbivores in certain populations. And if whole genome duplication is acting as a diversifying agent, we may expect insect herbivores to start going through the pathway of diversifying through processes such as host-associated differentiation, or through other things like that. So finally, if you'll allow me a moment of shameless self-promotion, I'm also giving a talk at Botany that's happening in about 2 weeks. So in today's talk, I told you about how herbivores are affected by feeding on neopolyploids. But if instead you're interested in looking at how the neopolyploids respond to herbivory, that's what I'll be talking about in my botany talk. So please come check it out if you're interested. And then similarly, my lab mate Thomas, who gave a talk a few weeks ago, is going to be talking about how neopolyploidy and Euchera cylindrica causes increased nutrient limitation phenotypes of functional traits. And his talk will be great. So I also highly recommend that you check it out. So finally, I would like to thank everyone shown on this screen and everyone listed for their help with experimental design and advice, as well as some emergency pupa collection. I couldn't have done it without them. And I again would like to acknowledge all of my co-authors who couldn't be here today. So thank you all so much for listening, and I'm happy to answer any questions.
All right, well, thank you for a wonderful talk. As usual, we will let folks post their questions to the chat window, and then I think as we've done the last couple of weeks, let people turn on their video and audio and just go ahead and ask those questions as well. So I'll give everybody a minute or two to put those in. I don't like to ask the first question, but I will. Oh, there we go. All right, Peshol has the first question. And if Peshol wants to fire up their video and ask away, that also works. or go ahead and answer it, I suppose. So what hypothesis, yeah, if any,
Yeah.
There you go.
So the question is about if what hypothesis, if any, can be proposed for the observation that cross-generational effects had a significant impact on herbivore performance. So in this system, I would say that cross-generational effects really didn't alter herbivore performance. It was more based on the ecotype or the geographic origin of our plants. And from that, I more take that it's just selection acting in a population that can change different traits in it. That's what I would guess. Is it okay if I go to Robert's question?
Yeah, I think the next question there is from Rob, and if Rob wants to turn on his video too and ask in person.
Sure, I could do that. Thanks, Mike.
Yep.
A lot, and for your talk, it was really great. You mentioned that you were using this generalist herbivore for your assays here. I'm wondering what you would expect for a specialist or something that might be more of a specialist herbivore on these plants, or any plant really, I guess.
Yeah, that's a question I'm actually really interested in. That's one of my proposed PhD chapters. So my thought would be that a specialist herbivore would perform best on whatever its natal host cytotype would be. So in this instance, I think it would perform best on if it's in some of our populations, diploid Arabidopsis, but if it was in our established polyploid population, then you might expect them to perform best on that established polyploid. That's what I would expect.
Cool, thank you. I think the next question is from Tia Lynn.
I can read my question, but very nice talking, Anne. I'm curious, I'm always impressed with the differences among genotypic lineages for the effect of polyploidy. And so I'm curious, and you have it in your system as well. So I'm curious if you have any ideas of sort of what traits might underlie those genotypic differences. And
yeah, thank you. That is also a question that I'm interested in. So I think I still have my screen showing, right?
Yes.
So what I'm particularly interested in are these herbivores which are feeding on Prudka, because they're always opposite. And what was really cool during the experiment, it was of course blind, so we didn't know which ecotype was which, but we later found out in Prudka, the herbivores, when they were feeding on the rosette, they would eat the older leaves and they would skeletonize the leaf, so they wouldn't eat the veins of the leaf. But when they got to the top rosettes, like the top 2 rows of leaves, the newer leaves, they wouldn't eat them at all. Like they would rather starve or try to eat the transparency than eat those leaves. So to me, that suggests that those plants from Prudka must have a lot of secondary metabolites in them. And that's something that I'm really interested in looking at to see if that's true. But then I'm also going to look at nutritional content to see if maybe that's affecting it as well.
cool. Could you also tell us a little bit about the established autotetraploid? Was that like a lab escapee, or is it natural tetraploid that occurred in nature?
That's a great question. I believe it's a naturally occurring established tetraploid, but I'm not 100% sure on that.
Thank you.
All right, we have another question from Mark Ritchie.
Hi, Mark.
Yeah, hi, Anne. You found this signal of the geographic origin of the polyploid as having an influence, either positive or negative, on the herbivores. And so what do you, I mean, in terms of like knowing where these geographic origins were, can you speculate on why you think the ones from certain sites supported herbivores and the others did not?
Mm-hmm. My initial guess would be that the diploids from those sites, for example, if it was a site where they're not really attacked by herbivores a lot, so there's low herbivory pressure, maybe they just don't naturally produce as many secondary metabolites, and maybe that would be also expressed in the polyploid. That would be my best guess. Again, it also could be differences in nutritional content, and that's something I hope to measure as well.
Okay, but I guess what I'm trying to get at is why would you expect there to be geographic variation in herbivory on Arabidopsis? Is it driven by thinking about the environment, or biogeographic history or something like that. I realize you may not know all of that. I'm just, that's use the word speculate.
Yeah. Huh. I'm not sure.
Yeah, one of the things that I didn't have a great memory of your map, but I mean, it's clear that there's a lot of climate variation across, particularly as you go from southwest to northeast in Europe. You're basically getting colder, shorter growing season days. You also get into regions in which cabbage is a widespread crop, such as in Eastern Europe, as opposed to in France, where my guess is it's not. And so I'm just wondering if you looked at that kind of background, whether that makes any kind of sense with your data.
I did not, but that's definitely something I should look into. I'm gonna write that down.
All right. Well, thank you, Mark and Anne. Looks like we have one more question here from Jeanette Witten.
Hi, Anne. Really cool system. I was curious about whether or not you know, or whether or not you have any inkling about whether endopolyploidy is part of the plant's response to herbivory in this context, and whether or not that's something that you're planning to look at or have looked at.
Yeah, Arabidopsis is definitely a big species with endopolyploidy studies from Ken Page. I haven't personally looked at any of these ecotypes and compared endopolyploidy, so I don't know. We do use Call and Landsberg, which I talk about in my botany talk, which Ken Page uses, but I think we use a different accession than he does.
Okay.
So the short answer is I'm not sure, but it could definitely potentially, I think, impact responses.
Cool. Yeah, I wondered about that Prudka one and whether or not it has some different feature and whether that could impact. Awesome. Well, I'll look forward to your botany talk.
Thank you.
All right. I had one real quick question as well to follow it up, and thank you, Jeanette, for that great question too. My question is, do you see any difference in trichomes or any other sort of structures that across these ecotypes? I'm just sort of curious if there's any, not necessarily chemical defense related, but physical defense differences that may be playing a role here.
I admittedly, I haven't looked for my specific ecotypes, but it is something I plan to look at. I do know in Arabidopsis, though, there can be differences. So I would guess there probably is, but I'm not sure.
Cool. Excellent. Well, thank you.
Thank you.
I look forward to the talks at Botany as well. And yeah, that's 2 weeks away. And I think everybody's scrambling to get their videos recorded and uploaded this week. So all right. Well, with that, we'll switch over. And if you could go ahead and relinquish control, there we go. And then I believe Sílvia is coming to us from Northern Arizona, it appears, with the background there, or Southern Utah.
Maybe I don't like it very much, but I actually not certain that I will be able to switch it.
There you go. Well, it looks pretty good for me. All right.
Okay, so I can continue. Why not?
That's right. Well, I'll go ahead. So our next speaker is Sílvia Castor. She's currently an invited assistant researcher at the Center for Functional Ecology at the University of Coimbra in Portugal. And her research interests focus on flowering plant reproductive biology, plant pollinator interactions, and plant ecology in general. Although she started working with biology and conservation of endangered species, during her postdoc in the Czech Republic, she started working with polyploid complexes and became interested in species interactions at contact zones and the ecological processes driving their current distributions. And today she's going to tell us about work that covers that range of exciting topics. Can we live together? The role of multiple reproductive barriers enabling cytotype coexistence in a tetraploid-octoploid contact zone. And I'm really looking forward to hearing this talk as well. And with that, I'll let Sílvia take it away.
Thank you very much. Thank you for the introduction, and also thank you very much for organizing these wonderful webinars, which enable us to learn a lot and at the same time to present our work. So, can we live together? That's the topic of today. And actually this work has been developed within the PhD thesis of Sílvia Castor that you see here in the corner on the evolutionary ecology of polyploids. And I don't need much introduction. So polyploid is a widespread phenomenon in the evolutionary history of flowering plants. And it has been proposed as an important mechanism of sympatric speciation since it results in immediate reproductive isolation. So polyploids arise through the fusion of unreduced gametes, but the intriguing part is how do polyploids establish in the parental populations? So the models predict that mixed ploidy populations are rare and unstable, and evolutionary unstable, because frequency-independent selection will eliminate the minority cytotype. This is mostly because under random mating, the tetraploid will produce mostly unviable offspring, triploid offspring. So to overcome this minority cytotype disadvantage, the polyploid will need to have an advantage in comparison with their parentals, or alternatively, it could disperse elsewhere to escape competition with parentals. So indeed, one of the mechanisms promoting polyploid persistence in nature is ecological differentiation. And we have research that founds ploidy-mediated effects, not only on morphology and breeding system, but also on ecological tolerances that may enable the neopolyploids to colonize areas that are not occupied by their parentals. Still, in many cases, cytotypes form contact zones where they occur in close proximity and occasionally form mixed ploidy populations, as the one you see here in the corner. And actually, as we screen more natural populations, we see that it's more frequently to find a mixed ploidy population than expected. And again, also, this spatial proximity creates the potential for ecological interactions between the cytotypes. So, mixed ploidy populations are actually possible in nature and are possible when a set of biological attributes, such as plant traits that promote assortative mating, or larger viability and fertility by the polyploids, or recurrent polyploid formation through the production of unreduced gametes, can ameliorate the fitness disadvantages of a rare cytotype. And within the barriers to intercytotype pollen flow and to mating, we have several barriers that can act isolatedly or in concert to promote assortative mating. And this could be habitat isolation or temporal, behavioral, or mechanical mechanisms preventing pollen flow. Or if pollen flow occurs, then we may have gametic interactions that ultimately prevent intercytotype mating. So in the literature, we have several examples of polyploid complexes where reproductive barriers have been studied. Still, a comprehensive estimates of reproductive isolations across multiple barriers are less common or restricted to a few polyploid systems, although they are key to understand the dynamics of the contact zones. Thus, our main objective was to quantify the strength of isolation across multiple reproductive barriers in a tetraploid-octoploid contact zone to try to understand the potential for cytotype coexistence. And for this, we used the Gladiolus communis polyploid as study system. This is a Vitranian putative autopolyploid, mainly composed by tetraploids and octoploids, although hexaploids have also been reported for the Iberian Peninsula. Also, previous taxonomic treatments recognize several taxonomic entities, but recent morphological and molecular studies failed to support them, and the complex is seen as an aggregate. So surveys in the Southwest Iberian Peninsula using flow cytometric analysis of over 100 populations show us a complex contact zone. You can see here in the maps, in white the tetraploids and in black the octoploids, in gray the hexaploids. And we can see that we have two dominant cytotypes, the tetraploids and the octoploids, occurring mostly in pure ploidy populations. You can see here the proportions, but we also have 13% of mixed ploidy populations with variable cytotype compositions. Also, we have hexaploids occurring inside the contact zones, outside the contact zones, and even forming pure ploidy populations in some places, as you can see here. So additionally to this, it seems that they can co-occur together, or they co-occur together. Additional to this, modeling of niche suitability using environmental variables also suggests that the distribution of the dominant cytotypes, meaning tetraploids and octoploids, cannot be explained by environmental differences. So we are left with reproductive barriers. And in this case, we quantify the sortative mating through flowering, differences in flowering, differences in pollinator behavior, differences in flower morphology that could affect pollen deposition on pollinator body, and as post-pollination mechanisms that act after pollination, we studied the role of self-fertilization and gametic competition. And for that, we went to the contact zone and select 3 pairs of tetraploids and octoploids populations, and we studied them in natural conditions and also at the common garden. So starting by flower differences. So phenological isolation is determined by the flowering time overlap and affects the probability of pollen exchange between cytotypes. So we monitored the number of open flowers per cytotype a long time, and actually observed a high degree of overlap in flowering times between the two cytotypes, both in natural conditions and in the common garden. So this doesn't prevent pollen flow. So the next step was to study pollinator behavior, and this is because some morphological and physiological differences in flower characters may influence pollinator foraging behavior. And to study that, we built experimental arrays with tetraploid and octoploid plants and monitored pollinator behavior to ultimately calculate flower preference index and floral constancy index. And we can see here the main pollinators of these cytotypes in the studied populations, and these are generalist insects that visit the flowers. And we can also observe that most of them show no preference for a specific cytotype, here denoted and statistically not significantly different from 0.5, revealing no preference for a given cytotype. And we also see random foraging behavior for most insects, meaning that they just switch between one and the other randomly, except one that shows an alternating foraging behavior, that is Antidium. And we think that is most probably because it tends to visit the closest inflorescence, so it alternates actually between cytotypes. So consequently, pollinator behavior also does not prevent intercytotype pollen exchange. But still, flower morphology can also determine the pollen removal and deposition on the pollinator body. And this has been studied in other species complexes and is rarely addressed in polyploid complexes. So for that, to try to investigate that, we measure the morphology of the morphological overlap between tetraploids and octoploids, quantifying the length of anthers and overlapping them of tetraploid anthers and overlapping them with octoploid stigmas and vice versa. And for each individual, calculating actually the number of individuals of the other cytotype that overlap with it. So already in the field, it was very clear for us that tetraploids and octoploids are very similar floral morphologies, we are not able to tell them apart. And in this figure highlighted in gray, you can already readily see that the sexual organs largely overlap between cytotypes. So, up to now, it is clear that pre-pollination reproductive barriers are very weak, and thus pollen exchange is possible. But if pollen exchange is possible, then gametic barriers can cause different searing abilities and can ultimately reduce intercytotype mating. Thus, we performed a set of pollination experiments, excluding pollinators and manipulating the sources of pollen, and quantified fruit and seed production, as well as the offspring of the ploidy level. And for that, we built two different kinds of pollinations, single ploidy pollinations and mixed ploidy pollinations. For the single ploidy pollinations, I will try to guide you on this. We tested self-incompatibility, treatment 1 here for both cytotypes. We also tested the outcross within cytotypes here, and this gives us the optimum fitness. And then we tested the outcross between cytotypes here. That will enable us to test crossability between cytotypes. So, in this graphic provides you the combination between fruit and seed production, and at the same time, it provides you the ploidy of the offspring obtained in each of the crosses. We can see that this is a self-incompatible plant, and it is quite interesting to see that although self-incompatible, the incompatibility is less strong in octoploids in comparison with the tetraploids. The within cytotype crosses yielded the expected results. So the tetraploids were obtained in tetraploid crosses, and octoploids were obtained in octoploid crosses. Sorry, I forgot to say the color scheme is always the same. Diploid for yellow for tetraploids, and blue for octoploids, and green for hexaploids. And the between cytotype crosses yielded different results. We observed a slight decrease in fitness, and at the same time, we observed high proportions of hexaploids being produced by both cytotypes. It is also interesting to note that we see a high proportion of octoploids in both crosses, which suggests the production of unreduced gametes by tetraploid individuals. So all these together suggest that there are some gametic isolation, but this isolation is not complete because the cytotypes are still able to still have some cross ability. So going for the mixed ploidy pollinations, we made two different treatments, and this was mainly because pre-pollination barriers are very weak. So mixed ploidy pollen loads are expected in mixed ploidy populations. So for the first we tested a mixture of outcross pollen from tetraploids and octoploids that will enable us to assess gametic selection against the alternate cytotype. So you can see here, so mixed between the mixed pollen loads, and the final treatment is an outcross between cytotypes plus selfing. You can see here and here. Okay, this will enable us to depict the role of selfing in working as a reproductive barrier. What do we observe? So in mixed ploidy, in mixed outcross, we obtained similar results as the within cytotype crosses, and this supports the occurrence of gametic selection against the alternate cytotype. So tetraploids produce mostly tetraploid offspring, and octoploids produce mostly octoploid offspring. When selfing was involved, the scenario was completely different, and it was different between cytotypes. So in tetraploids, the self pollen seems to block the formation of hexaploid offspring, while in octoploids, a high number of hexaploids are produced, and actually similarly to the between cytotype crosses. And this suggests that the pollen of the tetraploids is more successful in fertilizing these ovules than the self pollen, resulting in lower levels of reproductive isolation for the octoploids in comparison with the tetraploids. Sorry. So, can we sum up all these and provide some reproductive isolation values? Yes, of course. So from previous work, we were able to calculate geographical isolation values, which are still fairly high, as you can see here. But we have mixed ploidy populations in nature. So the other barriers will have to work together. And what do we have? We have low temporal isolation. We have low behavior isolation, low isolation due to pollinators' behavior. We also have low mechanical isolation. So the gametic interactions are the ones, the key barriers playing, or the barriers playing the key role in these interactions. How? Through selfing, especially in the tetraploids, where the selfing imposes a higher reproductive barrier, but also through different, through selection against the alternate cytotype that will depend on the pollen loads from being received by the stigmas of each cytotype. Okay. Overall, this results in high reproductive isolation levels for both cytotypes. And we were still not very happy because we were quantifying these barriers individually, and we questioned, does this work under real conditions? So to test it, we built an experimental tetraploid-octoploid population in the common garden, and ensuring similar conditions for all plants, similar cytotype proportions, and similar pollinator assemblages, and let nature do its job. And finally, we quantified fitness and offspring ploidy. So the results obtained were very, very nice because they were in accordance with the cumulative estimates based on individual barriers and show high reproductive isolation values for both cytotypes. Okay, to sum up, in sympatry, post-pollination gametic barriers were key barriers in this complex. And actually the strong gametic selection against the alternate cytotype may maintain these tetraploid-octoploid populations. However, pollen load composition actually determines the magnitude of reproductive isolation. And because pollen load composition determines not only the fitness and the offspring ploidy, but also because contact zones are characterized by different mixed ploidy spatial arrangements, the interactions between the cytotypes are expected to be very complex in natural contact zones. Also, I think it's important to highlight that the relative fitness of the octoploids may actually increase, not only because of the production of unreduced gametes, but also because of hexaploid production that could be bridges, and due to also high selfing success in comparison with the tetraploids. So actually, further studies in contact zones are needed to really understand the fate of these populations. Thank you very much, and I will be happy on addressing, answering to some questions.
All right, well, thank you for a wonderful talk, Sylvia. Like previously, if folks want to put their questions in the chat box, and then we'll fire up your video and have you ask.
Oh, let me see my bit.
We're not quite there yet.
Okay, I'm trying to.
All right, Rob's up first.
Como é que eu abro isto? I need my technical advisor.
No problem.
I'm struggling with computers today because everything is shutting down. I'm not able to see the chat.
All right, well, I can help.
Can you ask it?
Yeah, I can. So Rob, I'll let you go ahead and ask.
Okay. Yeah, I just wasn't sure she could even see anything. Thanks a lot, Sylvia. That was really awesome. I guess with the variation that you see geographically structured in the makeup of those populations, where you have several mixed populations, and then you have these pure populations or areas that are pure in cytotype. I'm wondering if you guys have observed any indications or are currently studying whether you might have variation in that gametic selection that's occurring. So for example, there might be reinforcement occurring between the tetraploids and hexaploids where they co-occur. but in areas where they occur separate from each other, you might see lower levels of gametic selection?
That's very interesting question indeed. Yeah, up to now we have been working only in these contact areas, and actually we wanted to work really in mixed ploidy populations, not only in the common garden, and tried to test it, but they are also in environments that are really difficult to study because they are grazed and they have different all sorts of pressures. And I think you might be right. I think in other areas where we have areas dominated by a given cytotype, and then we see punctual appearance of exoploids, which actually appear in areas dominated by tetraploids, we could have different gametic selections indeed. But that's maybe the next issue to address, definitely.
All right, thanks. That was a great question. Looks like up next, the next question is Dirk Albach. Dirk wants to fire up.
Yep.
There you go.
Hi.
Hi.
Hi, everyone. I was wondering about the post-gametic selection that you may encounter. Did you measure the fertility of the offspring?
We tried to germinate the seeds. We really wanted to do stuff with the offspring. We tested a lot of germination trials, and we were not able to make them to germinate. So that's unfortunately with this plant, we need to scarify them, submit them to cold conditions. So we were not very successful on that, and that's a pity because I really wanted to pick, especially the exoploids. I'm very curious about these exoploids, and we really wanted to do more stuff with it, but we weren't able to germinate the seeds. I don't know if maybe we need to switch it to natural populations and germinate them in natural conditions and follow them through years. Maybe that's an option, but in the lab we struggle a lot.
Okay. So you have the nicer flowers, but the problems of Arabidopsis.
Yes. We always have a problem with the system. It's never perfect.
Great, thanks.
Thanks.
All right, up next is Tia Lynn.
Hello, that was really nice talk.
Hi, thanks.
Oh, did I just? Am I still here? My screen just went totally blank. Okay, my screen went totally blank, so okay. I was curious about the results where self-pollination modified the impact of the other cytotypes pollen, and so I'm curious like how much if they have autonomous autogamy or if that varies amongst the cytotypes?
So autonomous autogamy, you say that they spontaneous self.
Yeah.
You mean that? Okay. So here we manipulate every cross. Okay. So we emasculate all the flowers, and the selfing was made by us. Okay. So we didn't test it autonomous selfing, but from bagging, from experiments of flower bagging, we have no output from that. Usually there is no offspring being produced. So I would say that both are not able to self-pollinate alone. They are able to self-pollinate if pollinators promote between flower pollination or within flower pollinations. Did I reply to the question? Or maybe the second part not? But I think we lost her.
I think Tia Lynn, yeah, we might have lost her. She seems to have frozen.
Yes.
Okay. Thank you.
No.
Okay.
Thank you. Okay, well, up next, next question then looks like from Jeanette Whitten.
Yeah,
Jeanette,
there's a note.
Hi, Sylvia. Such a cool system. I love this work. Congratulations.
Thank you.
I was wondering, and this is a little bit speculative, but do you think that coexistence is stable in this system? So are the forces you see like strong enough that they would likely drive further divergence either in reproductive traits or in ecological traits?
I think that based on these results, if the proportions of the cytotypes are fairly similar, the populations, the mixed ploidy populations may be maintained for some time in nature. Still, I think there are, I don't know how to say it in English, but there are several issues that are still bothering us, like the ones I finished in my talk. And I don't know how much they contribute to the stability or instability of the population. And I would like very much to quantify that as well. For example, seeing the tetraploids producing such amount of unreduced gametes, at least detected in the offspring, that is very surprising, and they will be contributing to the octoploids directly. If they are receiving mixed pollen loads, they will definitely contribute directly to the octoploid population. So maybe in short term, the populations may be maintained. Also, this is a bulbous plant, so they can maintain in the population for a long time. We didn't found in the common garden, we didn't found differences in the asexual reproduction, for example. Bulbs are fairly of the same size. They multiply more or less in the same rate between years, between cytotypes, and we didn't found differences, although we didn't formally test it, okay? So the other contributions through unreduced gametes, even the exoploid production could also work as a bridge to feed the octoploid population. The fact that selfing yields higher offspring in the octoploids. So all these suggest that actually octoploids could increase in time within the population. I don't know other stages. I don't know if seed germination is different. I don't know if plant survival is different. If the dynamics of the population of the tetraploid individually and the octoploid by its side are different. So I say that yes, they can maintain stable for some time. I don't know in long term.
Yeah. It's super interesting that, to me, that there are so many populations where it's mixed tets and octoploids and no hexaploids, because that too suggests that the barriers are effective between those.
But we also have a lot of hexaploids. Am I still sharing the screen?
Yeah.
Yes, you are. Yep.
Yes. Okay. I can put in the graphic just to show it rapidly. Just a moment. So are you able to see something?
Mm-hmm.
Oh, Jesus. Okay, so you can see here in the map, I will project it because I can put the pointer. Okay, all these gray, for example, here actually are octoploid with exoploids. You can find several octoploids with exoploids. Mm-hmm. And we have also tetraploids with octoploids. Tetraploids with exoploids, and even to the southern part, I think we might have even more contact zones in these areas. Mm. So actually, I think only this shot, I think it's well representative. Then all these parts, I think we can find a mess, a complete mess.
Cool, thanks.
Thank you.
All right, looks like the next question is from Anne.
Sylvia, I think my question and Carrie's question are kind of similar. In your common garden, did you look at the spatial structure within populations, or were they more among populations?
Yes, we try to map the individuals within populations, and we selected mm-hmm. some populations to map it. We were not very successful in the ones we choose because then they were mourned and they had sheep eating the plants. But for example, here you can see some populations. For example, this is tetraploid-octoploid populations with fairly high, with fairly similar proportions between the two cytotypes, and you can see there is some distance. This is in meters here. So there are some distance, but they are actually clustered. Then you have here a tetraploid hexaploid population, and hexaploids are just mixed within the population. And here you have a very quite complex one, but it's dominated by octoploids. And then we can see hexaploids here, 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 here. And we have one tetraploid lost, and we even have a nonaploid somewhere, maybe this one, I think. But we were not very successful. We really wanted to map more populations like this one to see microhabitat differences. But yeah.
Thank you.
Yeah, and I think that question is, Carrie's next, but I think that question is pretty similar. Maybe Carrie has an addendum or addition that she wants to ask.
I don't know if I really want to necessarily follow up. I was just curious sort of if these microhabitat segregation could drive the, patterns as well, could sort of help facilitate reproductive isolation. And I know Brian Husband has shown that in his own work, just thinking about how intermixed they actually are. And
Definitely. And if the pollinators have no preference, they will try to move in the neighbor plants. So they will promote, if cytotypes are clustered, they will promote crosses within cytotypes instead of between cytotypes, definitely. And that was seen also in buttercup, for example, I think. Yeah, it could be.
Yeah, exactly. Yeah. I was just thinking that maybe, pollinators aren't necessarily going to pick up on the small differences between these things. So it would be logical that then they could still cause reproductive isolation if there is spatial segregation. So it's truly going to depend on the structure of local populations as to how important pollinators are.
Yes, I agree. Yeah, I agree.
Perfect.
Also, we measure lots of flower traits, and we were not able to say anything. That ones are only related with sexual organs, but we measure petals, sepals, petals, nectar production, all sorts of things, and they are just very similar.
Yeah, that makes perfect sense. So cool. Thank you.
Thank you.
That's a great question. Thank you, Carrie and Anne, for those questions about the structure. I have one last real quick question that sort of inspired me thinking about some of these responses. But when you think about the floral morphology, it seems like for a lot of these showy flowers like irises and things, like UV nectar guides and color patterns can be important. I'm always curious if people look for these, and if so, do you see any difference in that sort of hidden morphology that we can't see ourselves?
That's very, very, very good point. And I think we miss it in many studies. Maybe in some systems, they are not so important. Here, actually, this one has some patterns to guide pollinators to the entrance of the tube where the nectar is accumulated. We always think on that as well. And we, for example, in Gymnodenia conopsea, an orchid, we try to measure reflectance to see different patterns. In the end, we didn't saw any. In this, we actually just didn't measure it because we don't have the apparatus to do it, but it would be interesting to do it. Even nectar composition could be interesting. In sugars, we didn't saw any difference in percentage of sugars, but who knows other traits that could maintain the fidelity of the pollinator or something. But again, as Carrie said, if they are not able to tell apart either by, or even fragrance could be also a very important cue. But if they don't have a strong cue to detect the cytotypes, then the spatial arrangement actually it will be key.
And I remember,
although I would like very much that pollinators will play a different role. But
well, I also remember from Yuval Sapir's work on his irises that the bees will warm up. In the mornings and so the temperature of there was temperature differences between different species, and I was
they work as shelters, right? I think.
Yeah, right. Yeah. And I don't know if that sort of biology happens in this system or not, but
here the coral is quite open. So, and the visitors are really big, actually, and most of the main pollinators are big insects, and the pollen deposition is actually in the thorax in the upper part. And they don't fit inside they don't fit inside.
All right. All right. Well, thank you very much, both
you.
Anne and Sylvia, for 2 wonderful talks today. I really enjoyed both of these, and look forward to next week's, or talks, I should say the next webinar's talks in 2 weeks, which will be from 2 graduate students, Justin Conover and Heather Gillette, a little bit more in a genetics direction from this week's more ecological focused and interaction focused talks. With that, I'm going to wrap up our recording here and invite people to stick around and chat about polyploidy and whatever else they want to talk about today. But again, final thank you to both Anne and Sylvia for doing great talks, and put a plug in for everyone to check out the talks at Botany 2020 online. I assume some of those will be made available at some point later. I don't quite know how all that's working, but for those of you that are presenting and participating in the meeting, hopefully it goes well and should be a good series of talks on polyploidy as usual at Botany. All right, well, with that,